Jane Doe is a 5-year-old child with kidney disease. Her kidneys are no longer functional, and so she's on dialysis, hooked onto a machine that acts as her artificial kidneys, similar to the one on the screen, but she's on this 3 times a week, 4 hours a day. Fortunately, she receives a kidney transplant, which currently is the only treatment that can provide her with a lifelong solution to her disease. But is it? Long-term transplant survival rates remain poor, where in BC, only 1/3 of transplants are functional by 10 years. Jane will likely require multiple transplants throughout her life, with no guarantee she'll get one, as the waitlist for a transplant is over 400 patients long. She will return to dialysis, which has a 5-year survival rate of 44%. The majority of long-term transplant failures is due to rejection, and this is when the recipient's immune system sees the donated organ as foreign and attacks it. As researchers, how can we prevent rejection from taking place? How can we prevent the immune system from being able to differentiate between a host kidney versus a donated kidney? Well, it turns out there is a way. Studies show that transplant recipients who are well matched to their donors at particular genes result in significantly better outcomes. As part of my thesis, I studied the impact of matching at these genes in BC kidney transplantation. One of the challenges with matching is that it's not feasible, and this is because the genes we want to match at are very complex, as there are over 20,000 genetic variants. In my research, I used the power of DNA sequencing to study these genes in BC kidney transplantation. I also used bioinformatics to analyze and compute these DNA sequences, and this allows me to study the structural biology of the proteins that these genes encode. Through these tools, we now know that there are key regions on the exterior of the protein that the immune system targets in rejection. We can think of this as the protein being a handful of assorted M&Ms, and you're the immune system. You only like eating the red ones, so you only target those red ones, ignoring all the other colors. The immune targets are these red M&Ms. In my research, I showed that there are only 150 of these immune targets in our BC cohort, greatly reducing the complexity for matching. We are hopeful that these results show that matching is feasible, and this will change the way BC allocates its organs. Currently, my lab is working with Canadian Blood Services to incorporate matching into a national organ sharing protocol. At the end of the day, we want to better match patients to their donors so that individuals like Jane can keep her transplant for life.